Prior to performing this experiment, we all made sure to dress appropriately with safety goggles, gloves, and proper clothing. Made sure to review our pre-lab notebooks with information on safety and waste disposal. Once adhering to these safety guidelines, we dived into our first dilution experiment. First, we measured out 1.25 volumes of copper sulfate pentahydrate, which has the appearance of resembling blue crystals and serves as the salt in this experiment. By collecting them in a watch glass, we used weighing paper to transfer it into a beaker to be dissolved. In order to aid this dissolving process, we rapidly swirled the beaker until all of the salt was no longer visible in the solution and had the appearance of a light blue color. Next, we adhered to proper technique by moving to the hood to add 5 milliliters of ammonia to develop our solution. Once again, to aid this dissolving process, we adhered to proper technique by rapidly swirling the beaker. Once we achieved getting the solution to change from light blue coloration to dark blue coloration, we transferred our solution into a volumetric flask.
During this process, we were initially adding exactly 10 drops of ammonia and began to add water after this process. At the halfway point before the meniscus, we evaluated what our solution gave a cloudy appearance, which would indicate the solution undergoing precipitation. Our solution did not become cloudy with the addition of water, so we continued to add water until the solution was in line with the meniscus. After completing these steps, we successfully obtained our stock solution for our dilution experiment and are ready to start the dilution process. Next, we made sure the mixture was fully mixed together by inverting the volumetric 2 to 3 times.
Finally, we transferred our stock solution from the volumetric flask into a clean, dry beaker to prepare for the dilution process. After successfully performing the transfer, we maintained proper lab procedure by being sure to rinse the volumetric flask 2 to 3 times and properly dry with a paper towel. Additionally, we gathered 3 additional beakers to hold each of our dilutions. Then we adhered to appropriate technique to perform our first dilution by using a blue bulb and pipette to transfer a small portion of stock solution back into the volumetric flask and a small amount of water up to the tick mark of the 25 milliliter volumetric flask. After successfully performing the first dilution, we repeated this procedure for the subsequent 2 dilutions. For instance, in regards to the formation of the second dilution, we took a small amount of the first dilution and diluted it into a small amount of water. Then for the third dilution, we took a small portion of the second solution and diluted it into a small amount of water as well. Through this process of obtaining dilutions from stock solution, their differences were highlighted. For instance, a stock solution has a known concentration and volume. Dilutions have a known volume, but do not have a readily known concentration. So, we have to use the dilution equation in order to solve for this unknown concentration value of the dilutions.
After successfully obtaining our dilutions, we went back to the hood to gather our blank solution of water. We adhered to proper lab technique by using a pipette to transfer the water from the beaker into a cuvette so that we can evaluate solutions and dilutions in the spectrophotometer. Through this process of obtaining dilutions from stock solutions, their differences were highlighted. For instance, a stock solution has a known concentration and volume. Dilutions have a known volume but do not have a readily known concentration. So chemists have to use their dilution equation in order to solve for the unknown concentration values of the dilutions. The dilution equation is C1V1 equals C2V2. Therefore, since the concentration and volume values are known for the stock solution, these values can take the place of the C1V1 variables. Additionally, since the volume of the dilutions were properly recorded by the chemist, these values can take the place of V2 variable. Since obtaining these values, the chemist can use the other values in the dilution equation to solve for C2. Next, we gathered 4 additional pipettes and cuvettes to properly transfer our stock solution and 3 dilutions for evaluation in the spectrophotometer. At the spectrophotometer, we made sure to maintain proper lab technique by ensuring that there was no excess water on the outside of the cuvettes by wiping them with a chem wipe before placing them inside the machine. After performing our experiment and obtaining our calibration curve, we performed the following calculations.